All right, welcome back to the deep dive, everybody. We're always talking about the coolest, most cutting-edge stuff in science, and today is no different.
Absolutely.
Today we're going to be talking about something super fundamental, so basic to how our bodies work, all the way down at the, cellular level. We're talking about mitochondria.
Mitochondria, yes.
Now, you probably remember these guys from, high school biology class, right? The powerhouses of the cell.
Right. ATP, energy production.
Yeah, making all the energy we need. But get this, and this is what I think is so cool, they're also huge decision makers when it comes to whether a cell lives or dies.
Yes.
And they even have these surprising connections to our immune system.
Yeah.
It's way more than just energy, right?
Way more.
It's a whole, cellular drama unfolding. And these mitochondria are right at the center of it.
Yeah.
Now, before we get too deep into it, though, I just want to remind everybody, please don't forget to like the video and subscribe if you want more content like this. Okay, so mitochondria, life, death, immune system. It's a lot, right? And we're going to really try to break it down and understand how these things are all connected.
Yeah.
And one of the key things we're going to focus on today is how mitochondria actually orchestrate these different roles through these tiny little gateways in their membranes called pores.
Pores, yep.
And we're going to be exploring how the opening and closing of these mitochondrial pores, these little gateways, is like a central control point.
Right.
Especially when we're talking about cell death and inflammatory signaling.
Exactly. And what's I think really interesting is we've got to think about the origins of mitochondria. Because that really plays into this whole story, and I think makes it that much more fascinating.
Oh, yeah.
Because, if you look way, way back in evolutionary history, mitochondria actually have this bacterial origin.
Really? I did not know that.
Oh,
yeah. So they weren't always part of our cells.
No. So it's this idea of endosymbiosis. So billions of years ago, this free-living bacterium got engulfed by another cell, and instead of getting digested, it stuck around.
Wow.
And they developed this mutually beneficial relationship. So over time, that bacterium became what we know today as the mitochondrion.
So they were like, hey, wait, don't eat me. I can make you energy.
Exactly.
Wow, that's amazing.
So, but thinking about that means the internal components of mitochondria, their own DNA, their own proteins, could actually be seen as foreign invaders.
Right.
By our immune system if they were to, escape into the rest of the cell.
Oh, right, because our immune system is always on the lookout for anything that doesn't belong.
Exactly. So thankfully, under normal circumstances, all those potentially immune triggering components are safely locked away.
Okay.
Tucked away behind those mitochondrial membranes.
Right.
So keeping those membranes intact is super, super crucial.
So it's like a delicate balance, right? You need to keep things contained, but you also need those pores to open sometimes for those signals.
Right. And what's so remarkable, I think, about this whole system is that cells can actually use the controlled opening of these pores as a way to sense stress.
Oh, interesting.
And then trigger the appropriate response. So, if there's too much damage, it might say, okay, it's time to initiate cell death.
Right. Time to self-destruct.
Or, maybe release some signals to tell the immune system, hey, something's going on here.
Right. Come check this out.
It's really an elegant system, how it all works.
Okay, so we keep mentioning these mitochondrial membranes, right? MEM. Can you give us a little more of a visual? What are these membranes actually like?
Sure. So imagine a mitochondrion, right?
Yeah.
Kind of like if you've ever seen pictures of them, like a little bean-shaped organelle.
Right.
And it's got these two layers of defense. So on the outside, you've got the outer mitochondrial membrane, the MOM. That's your first barrier. And then tucked away safely inside is the inner mitochondrial membrane, or the MUM.
Okay.
But these aren't just, two simple skins. They actually have very, very different personalities, we could say.
Personalities. Okay, I like where this is going. Tell me more.
So the MOM, right, the outer membrane, that's kind of like the easygoing one.
Right.
It's got these protein channels called porins.
Okay.
And one of the most well-known ones is called VAAC. And these porins basically act like fairly large doors, I guess.
Right.
Allowing, small molecules up to a certain size to pass through without too much fuss.
So the outer layer is kind of like a sieve then.
Yeah, you could think of it that way. So it lets the smaller stuff, in and out relatively freely.
Okay.
But now, if we go to the inner membrane, the MMOM, That's a completely different story.
Okay.
This guy's much more selective, almost impermeable, we could say,
Right.
to most molecules. And this really strict control is absolutely vital because it's what allows the mitochondria to actually build up those electrical and chemical gradients needed to generate ATP.
Right. That's the energy currency, right?
Yeah, the main energy currency of the cell. And to get anything across this inner membrane, the MIM, the mitochondria, needs these dedicated transporter proteins. So kind of like specific doormen checking everyone's ID, very carefully.
So it's like the inner layer is the real gatekeeper then.
Exactly, yeah. And it's not just about controlling what comes in and out. The MIM is also where the action happens, right? So it's packed with all this molecular machinery, and this is where you find all those proteins that make up the respiratory chain.
Okay.
So that's the system that actually produces ATP, and it's really amazing when you think about how this inner membrane, this MIM, is structured.
Okay.
Because it's not just a smooth surface.
Right.
It's actually folded into these really intricate structures called cristae.
Okay.
And these cristae, they kind of increase the surface area. So imagine like pleats in an accordion or something.
Okay.
It just allows more space for all those respiratory chain proteins and all those reactions to take place.
Right.
And these folds, these cristae, they also create even more internal complexity within the mitochondria.
Okay, so more compartments, more organization.
Yeah, exactly. So, like, the points where these cristae connect back to the main part of the inner membrane, those connections are super tight.
Okay.
So they create these separate little compartments within the mitochondria, almost like having different rooms, inside a secure building.
Right, each with their own, like, specific function.
Exactly. So you've got different things going on in different parts of the mitochondria, all carefully organized.
Wow. It's a lot more organized than I pictured, I'll be honest.
Oh, yeah. And, to keep all of this together, you also have these contact points.
Okay.
Where the outer membrane and the inner membrane, the MOM and the mammal, they come really close together. And these contact sites, they're super important. Kind of like these communication hubs where different molecules can be exchanged between the two membranes. And there are also a lot of essential processes, like, for example, the creation of new membrane components, which happens at these contact sites.
Oh, interesting.
And this whole network, right, of mitochondria within a cell, it's not static. It's actually really dynamic.
Right. I've seen those cool videos where the mitochondria are, like, moving around.
Exactly, yeah.
Yeah.
So they can fuse together. They can split apart. They're constantly changing their shape. And even those cristae, those folds in the inner membrane, they can remodel too.
Wow. So it's like a constant dance.
Yeah, it really is. And what's amazing is that despite all this movement, all this dynamic behavior,
right,
those membranes, they usually maintain their impermeability. And this is like a really big deal.
Okay, so why is that so important? Like, why do they have to stay so tightly sealed, even when they're, like, fusing and dividing?
Well, think of it this way. Those membranes, they're basically holding back a lot of potentially dangerous substances, right? Yeah. So, if they were to break down or become leaky accidentally, all sorts of things could go wrong.
Right.
So, for instance, you have these molecules called apoptogenic factors.
Mm-hmm.
And these are like really potent signals. They can trigger cell suicide, basically.
Oh, wow.
And if those were to leak out into the main compartment of the cell, called the cytosol, that could be really bad news for the cell.
Right, because then you'd have cells dying that shouldn't be dying.
Exactly. And then you also have these things called AMPs, which stands for damage-associated molecular patterns.
AMPs, okay.
And these are basically danger signals that alert the immune system, hey, something's wrong here.
Okay.
And so if those were to leak out of the mitochondria inappropriately, that could trigger unwanted inflammation.
Yeah.
So, like setting off a fire alarm when there's no real fire.
Right. It's like a false alarm, then your immune system overreacts.
Exactly.
Yeah.
So keeping those membranes intact, it's like maintaining cellular peace and quiet, basically.
Okay. So intact membranes equal a happy cell. But what about when these membranes do become permeable?
Yes.
You mentioned that this is a key event in cell death, right?
Right. So this is where things get really fascinating.
Okay, let's talk about cell death.
Yeah.
Because this is something that, it sounds kind of scary, but it's actually super important.
Well, it's essential. It's essential for life.
For life, yeah, exactly.
Yeah.
So maybe you can explain, why is cell death even necessary in the first place?
So, we think of death as a bad thing, but when it comes to cells, it's actually a really crucial process. So let's talk about apoptosis.
Okay, apoptosis.
Yeah. So that's a very specific type of programmed cell death, And it's essential for all sorts of things. So, like when we're developing as embryos, apoptosis is what gets rid of those webbing between our fingers.
Oh, wow.
So it's shaping our bodies.
It's like cellular sculpting.
Yeah, exactly. And then throughout our lives, it helps to keep our tissues healthy. So, cells that are damaged, cells that are old, cells that are infected with viruses, they get eliminated through apoptosis.
Right.
And this is really important for preventing cancer.
Oh, right. Because if those damaged cells keep dividing, that's how you get tumors.
Exactly.
Yeah.
And it's also super important for our immune system.
Okay.
So, when our immune cells encounter a pathogen, they can actually trigger apoptosis in those infected cells.
Right. To stop the infection from spreading.
Exactly. And then after an immune response, you have all these immune cells that are no longer needed, and apoptosis is what gets rid of them. So it's like a cleanup crew, basically.
So it's really this like tightly controlled process that's happening all the time in our bodies.
Right.
And when it goes wrong, that's when you get problems.
Exactly. So if you have too much apoptosis, that can lead to things like neurodegenerative diseases, like Alzheimer's or Parkinson's.
No.
And then, if you have too little apoptosis, that can contribute to cancer. So it's really about finding that balance.
Right, the Goldilocks zone of cell death.
Exactly. And so where do mitochondria fit into all of this?
Yeah, because we keep talking about these membranes, right? Like, how does that actually lead to a cell dying?
So when a cell receives those signals to undergo apoptosis, the mitochondria, they know it's time to get to work.
Right, yeah.
So they undergo some really dramatic changes.
Okay.
So they might start to break into smaller pieces. Those internal cristae, those folds we talked about, they can start to remodel. But one of the most critical steps, and it's often seen as like the point of no return, the irreversible commitment to cell death.
Oh, wow.
Is when that outer mitochondrial membrane, the MOM, becomes permeable.
Okay.
So think of it as like the outer defenses have been breached.
Right, the gate's open.
Exactly.
And then what? What happens when that outer membrane fails?
So when that MOM lets go, it basically releases those apoptogenic factors that we talked about earlier. So, like cytochrome C, SMAC.
Right.
And once these guys are out into the main part of the cell, the cytosol, they kick off this whole chain reaction.
Okay.
So it's like a domino effect, right?
Right.
And this chain reaction ultimately leads to the assembly of a protein complex called the apoptosome.
Apoptosome.
Yeah, that's a great name, right?
Very descriptive.
So the apoptosome, that's kind of like the executioner, and it activates these really powerful enzymes called caspases.
Caspases. Okay, so this is where things get really destructive.
Yeah. So caspases, they're basically like cellular demolition experts.
Right.
They go around and they chop up all these important proteins and DNA, and that's what leads to the dismantling of the cell in a very organized way.
Wow. So it's like a controlled demolition.
Yeah, exactly. And it's not just cytochrome C and SMAC that can escape when that outer membrane becomes permeable.
Right.
There are other mitochondrial contents, like AIF, for example, and even mitochondrial DNA, mtDNA.
Mitochondrial DNA, okay.
Yeah. So that can be released as well. And here's where it gets even more interesting, right? Because, if that mtDNA gets into the cytosol, that's a really potent danger signal, a DAMP.
Right.
And it can be sensed by a pathway called C-Ganstum Day. And this pathway, when it gets activated, it triggers inflammatory signaling.
So, wait, hold on. So a process that's designed to get rid of a cell cleanly can also cause inflammation. That seems kind of counterintuitive.
Yeah, it does. So, but it turns out that during normal apoptosis, those caspases that we talked about,
right,
they actually have a role in keeping that inflammation in check.
Oh, interesting.
Yeah. So they do that by breaking down certain components of that Ng-Gansen pathway. They actually can degrade CGAS itself.
Oh, wow.
And this results in what we call immunosilent cell death.
Immunosilent. Okay, so it's like a quiet and tidy disposal.
Exactly.
Yeah.
So the cell can be dismantled without causing this big inflammatory fuss.
Okay, that makes sense. But you mentioned specific proteins that form those apoptotic pores.
Right.
So who are the main culprits there?
So the primary builders of that apoptotic pore in the MOM are two proteins called BAX and BA.
BX and BX.
Yeah. So think of these guys as the key architects of that membrane opening.
Okay.
And their activity is super tightly regulated because you don't want these pores forming all the time, right?
That would be chaos.
Yeah, exactly. And this regulation is done by a whole family of other proteins called the BCL2 family.
BCL2. Okay.
So you've got different members of this family. So some of them are called BH3 only proteins.
Okay.
And these guys like BIM and B, they can directly interact with BX and BAG.
Okay.
And basically they tell them, okay, get to work. It's time to form a pore.
Right.
But then you have other members of the BCL2 family that are called pro-survival proteins.
Okay.
So these guys, like BCL2 and BCLXL, they act like the brakes.
Right.
So they bind to BX and BEAK and prevent them from forming pores. So you've got this constant tug of war.
Right. So it's like a very tightly controlled system.
Right, exactly.
So when BX and BEAK do get activated, what's actually happening at the membrane level? Like, how do these pores actually form?
So when BX and B get that signal, it's go time, they move to the outer membrane, the domain.
Right.
They insert themselves into the membrane, and then they kind of change their shape.
Okay.
And they start to clump together, forming these larger structures called oligomers.
Oligomers.
Yeah. So it's like a bunch of them coming together.
Mm-hmm.
And it's these oligomers that eventually create the pores.
Okay.
And these aren't just simple holes, right?
Right.
We're talking about something a little more sophisticated.
Okay.
So they're thought to be what we call toroidal pores.
Toroidal. Okay.
So imagine like a donut, right?
A donut. Okay.
Embedded in the membrane. So the edge of the opening is actually formed by both the BAX and BA proteins.
Okay.
But also the fatty molecules of the membrane itself.
Oh, interesting. So it's not just like punching a hole. It's like integrating into the membrane structure.
Exactly, yeah. And this unique structure has a lot of implications for what can actually pass through these pores.
Right, because different shapes, different sizes, different molecules.
Exactly. And we also know these pores aren't static. They actually can change their size as apoptosis progresses.
Oh, interesting. So they can start small and then get bigger.
Exactly. So, even a relatively small opening, just a couple of nanometers wide, is enough for those smaller proteins like cytochrome C to escape.
Right.
But we've also seen evidence of, like, much larger openings.
Wow.
Like hundreds of nanometers across. And in those cases, the inner membrane can actually bulge out.
Will bulge out.
Yeah, through the outer membrane.
Wow.
So it's a really massive change in the structure of the organelle.
So it's like the whole thing is getting rearranged.
Yeah, exactly.
Yeah.
And we also talked about that lipid, remember?
Oh, right. Cardiolipin.
Yeah, cardiolipin. So cardiolipin, and especially its oxidized form, it becomes more enriched in the outer membrane during apoptosis. And it turns out that this cardiolipin, it actually contributes to the formation of those apoptotic pores.
Oh, interesting. So it's not just the proteins, the lipids are also involved in regulating this whole thing.
Exactly, yeah. So those membrane lipids themselves, they're not just bystanders.
Right.
They're active participants in the process.
Okay. And you were talking about how BX and BA kind of team up to create these pores. How does that assembly actually work? Like, how do they actually come together to make these structures?
Yeah. So they start off by forming pairs. So these pairs are called homodimers.
Homodimers.
Yeah. So it's like two of the same protein coming together. And then these pairs, they join together to form even larger complexes. And these are called higher order oligomers.
Okay.
So like building blocks.
Right. Building blocks.
And these oligomers, they can arrange themselves in different ways within the membrane.
Okay.
So sometimes they might form lines, sometimes they might form arcs, and sometimes they might form complete rings.
Mm-hmm.
And the current thinking is that it's those arc and ring-like structures that actually form the pores.
Okay, so it's not just like a random clumping. It's a very specific structured arrangement.
Right. And the size of these structures, those oligomers, it might actually determine what gets released from the mitochondria.
Oh, that makes sense. So smaller oligomers, smaller pores, maybe only smaller molecules can get out.
Exactly, yeah.
Yeah.
So the smaller oligomers might create those smaller openings that allow, like, cytochrome C to escape, but those larger assemblies, they could potentially create much bigger holes, right?
Right.
So that might allow larger molecules like mtDNA to be released.
And we know mtDNA can trigger inflammation, right?
Exactly.
So the size of the pore is dictating what kind of response you get.
Right. And it turns out that BAX and BAK, they can even influence each other's assembly.
Oh, interesting. So they're like coordinating.
Yeah. So they affect how quickly that pore grows. They affect the timing of mtDNA release, which in turn affects the downstream inflammatory signaling.
So it's not just like they're forming these pores, they're actually controlling what gets released and when.
Right. It's like they're coordinating the exit of different cargo.
Wow. And it's amazing how these tiny molecular interactions, right?
Yeah.
Can have such big consequences for the fate of the cell.
Right. For the whole cell.
So we've been talking about the outer membrane becoming permeable, right? But you mentioned that releasing mtDNA also requires the inner membrane to become permeable.
Yeah.
So how does that happen? Like, what's going on there?
Yeah, so this is a really, really good question and still a big area of active research.
Okay.
But what we know is that typically the intermitochondrial membrane, the MIPTO-M, it becomes permeable, or at least to some degree, during apoptosis.
Okay.
But the exact mechanisms are still being worked out. So there are a few different ideas.
Yeah.
One possibility is that, once that outer membrane is compromised, maybe some factors from the cytosol, the main part of the cell, can actually get into the inner membrane space.
Okay.
Right? So the space between the outer and inner membranes. And maybe those factors can then activate other proteins in the inner membrane that can cause it to become leaky.
Okay.
So maybe even BHX and B themselves could be involved in that.
Oh, interesting.
Yeah. Another possibility is that, those stress conditions that trigger apoptosis, they might directly activate some proteins within the mitochondria that can open up the inner membrane.
Okay.
So there might be some kind of internal signaling going on there.
Okay.
And we also know that during apoptosis, the internal structure of the mitochondria, those cristae, those folds,
Right.
they undergo these significant changes.
Right.
And those changes are driven by proteins like TBID and OP1. And so it's possible that, as those cristae are being remodeled, that might actually weaken the inner membrane and make it more susceptible to becoming permeable.
So it's like the whole structure is getting kind of rearranged, and that might create some weak points.
Exactly. Yeah. And there's even an intriguing hypothesis that the structure of the cristae themselves might actually help prevent mtDNA release under normal circumstances.
Oh, interesting.
Yeah. So, it's like these folds are acting as a physical barrier.
Okay.
And there are studies showing that if you have defects in the proteins that are involved in forming those cristae, that can actually lead to increased inflammatory responses.
Oh, wow. So it's like a protective mechanism.
Right. And then finally, we can't rule out the possibility that the inner membrane might just be physically damaged.
Okay.
Maybe due to swelling within the mitochondria after the outer membrane becomes permeable.
Okay.
So, there are still a lot of questions about exactly how the inner membrane becomes permeable. But it's clear that it's a really important step in the process.
Right.
Especially when we're talking about the release of mtDNA and triggering those inflammatory responses.
So inner membrane permeabilization, still a little bit of a mystery, but we're figuring it out.
Right, yeah.
And you also mentioned earlier something called caspase independent cell death?
Yeah, CICD.
CICD. So how do mitochondrial pores fit into that? Because we've been talking about apoptosis and caspases, but this seems like a different beast.
Yeah, it is a bit different.
Okay.
So what's remarkable is that you can actually get cell death even if those caspases, those main executioner enzymes, aren't fully active.
Really?
Yeah. And this happens when you have this widespread permeabilization of the outer membrane.
Okay.
Driven by BX and BA.
Right.
So you get this massive release of mitochondrial contents, and that can be enough to kill the cell even without the caspases doing their thing.
Wow.
And that's what we call caspase-independent cell death, or CICD.
So it's like a different way to get to the same endpoint.
Right. But it's not exactly the same, though, right?
Yeah.
Because there are some important differences.
What are the key differences between apoptosis and CICD?
So in apoptosis, the cell is really carefully dismantled.
Right.
It's like a controlled demolition, right?
Right.
But in CICD, it can be a little bit messier.
Okay.
So the dying cell, it might not display the same eat me signals that tell the immune system, hey, come get me, I'm ready to be cleared away.
Oh, interesting.
Yeah. And because those signals aren't as strong, those dying cells might actually stick around for longer.
Okay.
And that can actually be a problem. And you also don't see the same kind of dramatic shrinking of the cell and DNA breakdown
Okay.
that you see in apoptosis.
So the cell still dies, but it's a different kind of death.
Yeah, it's a different flavor of death.
And you mentioned this can be more inflammatory.
Yeah, so CICD often happens more slowly than apoptosis.
Okay.
And because those eat me signals aren't as prominent, the dying cells can stick around longer, which can trigger a stronger inflammatory response. And there's another really important thing, right? Okay. So CICD can actually trigger another form of cell death called necroptosis.
Necroptosis.
Yeah. And this happens through the action of a signaling molecule called TNF.
TNF. Okay.
Yeah. So TNF, it can bind to receptors on the cell surface and initiate a whole cascade of events that ultimately lead to the cell exploding, basically.
Oh, wow.
Yeah. So it's a very dramatic form of cell death.
So it's like a chain reaction then. CICD can set off necroptosis.
Exactly, yeah.
Yeah.
And so you get this amplification of the inflammatory response.
So in this case, those BAX and BA pores, they're not just leading to cell death, they're also really kind of setting off these inflammatory alarms.
Yeah.
That go beyond just the C8 pathway that we talked about before.
Exactly, yeah. So they're having these really broad effects on the immune system.
Okay. So it's really interesting how you can get these different outcomes, right? Depending on whether the caspases are fully engaged or not.
Yeah.
Right.
So the caspases are really key players in determining how the cell dies and what the consequences are.
Okay. Now, let's shift gears a little bit and talk about another type of mitochondrial pore that you mentioned, which is the permeability transition pore.
Or MPTP.
Yeah, the MPTP.
So what's the story with that one?
So the MPTP, this is another really fascinating player, and it's found in the inner mitochondrial membrane,
Okay.
the MIM. And it's known for causing this really abrupt increase in the permeability of the MIM when there's an overload of calcium inside the mitochondria.
Oh, wow. So it's like a sudden opening.
Yeah, it can be very sudden. And unlike those BXX and BA pores, which are, more selective,
Right.
the MPTP, it's relatively non-selective. So it allows all sorts of ions and small molecules to pass through.
Oh, wow. So it's like a floodgate.
Yeah, you could think of it that way. And I think you mentioned this earlier. The exact molecular structure of the MPTP is still a little bit of a mystery.
Right. It's kind of like a black box.
Yeah, it's been really challenging to nail down exactly what it looks like.
Right.